It is now my distinct pleasure to introduce today's moderator, Christopher Lee, who is a MD, PhD candidate at the University of British Columbia in Vancouver, Canada, and has a background in neuroscience and tissue engineering. He completed his bachelor in biology at the University of Victoria in Canada in the lab of Stephanie Willard, working on pluripotent stem cell-based neural tissue models. He then moved to UBC in Vancouver, where he completed his master in science in neuroscience under the supervision of Dr. Hakon Nygaard, where he's now performing his PhD research with a focus on developing 3D patient-derived pluripotent stem cell-based neural tissue models of Alzheimer's disease for investigating disease mechanisms and therapeutic screening. Christopher has contributed to several peer-reviewed publications and also received multiple academic awards, including the Jamie Castles Undergrad Research Award and the BCR Reg Med Trainee Exchange Award. And he is committed to translating advances in vitro disease models into clinically relevant insights. And with that, I will pass it on to you.
Thank you so much for the introduction. So I'll be moderating this talk. Really, that'll just be, getting the questions together and that you're providing and then giving them to Stefan. So I'm really going to jump now to another introduction because we're going to start the talk right away here. So I'll introduce Stefan now. So Stefan is a postdoc in our research lab. He's really a shared postdoc between the labs of Dr. Brian McVicker and Dr. Håkan Nygaard. In 2017, he moved here, previously working with Professor Helmut Kettenman at the Max Dolbrich Center, where he characterized microglial electrophysiological properties in different pathological models, including Alzheimer's disease. And when he came here in 2017, his focus was really oxidative stress and microglia interaction, animal models of Alzheimer's disease. And over time, he moved more towards these iPSC models that the Nygaard lab focuses on. And through that work, built this neurosphere model, which has been a major focus of our research lab. And his big focus here has been, microglia biology, iPSC-derived human models, and he's received many citations, published a lot of first authored papers, including the one that this bioprotocol paper is based off of, and the Journal of Neuroinflammation. And with that, I will hand it over to Stefan so we can start the talk.
Good morning, everybody, or good evening from wherever you log in. Thank you, Renee. Thank you, Chris, for the nice introduction. And I want to mention Chris is a co-first author to the bioprotocol paper. So he deserves some credit there. So yeah, I'm really happy to present our work, our protocols on these iPSC-derived neurospheres. And of course, the bioprotocol paper is the core protocol of just getting the physiological neurospheres, which have neurons, astrocytes, and microglia. But I want to talk, and I'm going to go over that protocol with you. But I want to talk about a bit more about then how we use that for disease modeling, particularly Alzheimer's disease. And then at the end of the talk, go through the unpublished stuff, basically the next generation of that protocol. And I'll talk a bit about why I moved on a little bit from that protocol. And yeah, show you that's more like a medley of different things at that stage, but I'm happy to present that unpublished work too. And yeah, let's dive right into it. So that's the webinar structure. I'm going to start with the bioprotocol protocol. So the step-by-step things you have to do to go from iPSCs to neuroprogenitor cells to then neurospheres, and then the parallel differentiation of microglia, how to get them to infiltrate into these neurospheres, and then showcase a bit the functional properties of these neurospheres of the microglia too, and show some staining examples. So, but that's just kind of the core part. And then this is basically what where the protocol was first published in this Journal of Neuroinflammation paper, where we used these spheres to develop a amyloidosis model for mimicking Alzheimer's disease-like pathology, and then look at neuroprotective properties of microglia in these cultures. We did a lot of sequencing, single nuke sequencing, and identified a lot of AD-like gene expression profiles, particularly when you add microglia. And I'm going to go a little bit over that. And then the last part, as I said, is the upscaling part, where because about 2 years ago, I started moving on to trying to attempt to scale these cultures up, and it's been quite a interesting journey. But it worked out pretty well, and I'm happy to showcase a bit of that. So basically go over that kind of newer version. We use both in the lab now, and both have their advantages and disadvantages, like the old protocol and the new one. And I'm going to describe that to you guys. And so first of all, I'm going to give a bit of an overview of this human iPSC-derived 3D neural tissue culture world. And of course, it all started with the development of that iPSC technology from Takahashi in 2006 and 2007. So that's been now 20 years, and this really opened up this whole possibility to make human model systems. Before you needed embryonic stem cells, and the ethical barriers to get this kind of material to start a model system are obviously pretty high. So this now allows you to take a blood sample, a fibroblast sample from a healthy patient, a diseased patient, and then make these stem cells and then differentiate those into neural cells and make your models based on that. And there are numerous different strategies. They all have their pros and cons. So there's no golden technique here. In terms of neuroscience, really one big hallmark paper was Mellon and Lancaster's 2013 paper on the first cerebral organoids. So these were kind of the first breakthrough 3D big neural cultures where you get this cortical lobe formation. We do organoids in the lab too. This is an image from our PhD student, Declan Brennan. These are far larger than my neurospheres. And I started off with organoids, actually, and there are a couple of reasons why I switched to these neurospheres. They're less complex, so they don't grow these cortical lobes. You don't get cortical layer formation, but they offer a lot more experimental control. And they are smaller, and especially when you want to study immune cell function and focus on microglia cells, that's a huge issue because the diffusion of media through an organoid is poor, so you have a lot of necrosis in the core, and it's incredibly difficult to control. So in these cultures, especially these spheres, you will not get natural microglia just differentiating. In some organoids, you might get microglia-like cells, but it's much easier to kind of differentiate them in parallel. There are multiple protocols available now, and then kind of let them infiltrate. But if you have a lot of dead tissue in the center, they will go for that dead tissue and deal with it. And then, so that's been kind of a problem when I try to work with organoids and microglia together, and that kind of made me shift to these more simple spheres, but we were actually surprised how complex they are. So that's kind of the background. And this is the protocol overview from the bioprotocol paper. It's a lot, and I'm going to guide you through the steps here. So the core part is, of course, you have human iPSCs, and you need to differentiate them into NPCs and into macrophage and then microglia. And you do that separately, and then you go through that neurosphere generation protocol, and then later add the microglia. So let's go step by step. The first thing you have to do is you need neural progenitor cells. So we have a protocol we use for several years now. It's a fairly straightforward dual SMAD inhibition protocol, which pushes those iPSCs into a neuroectodermal differentiation. So this will rapidly differentiate them into neural precursor cells, and those have the capacity to differentiate into neurons, into OPCs even, and into astrocytes, but not into microglia, which are of mesodermal origin. And the protocol is roughly 3-week long. It's a bit annoying because it involves daily feeding. So you go through this phase of very dense iPSC cultures, which you then differentiate into NPCs, and they start forming these neural rosettes. That's when you know, okay, you're getting NPCs. And that would be kind of after that, at the end of that protocol, and this NPC differentiation is also part of the bioprotocol paper, by the way. You end up with your P0 NPCs. You can use those directly to start neurospheres. What we actually do and work better for us is actually expand those with 1 or 2 passages. They mature a bit, And like a P2 NPC should look like this. You have this kind of small kind of rosette formations. You get some kind of neural processes where the density isn't as dense. And that's kind of your perfect material to start your sphere formation. And those ones you can freeze down. So that's the nice thing. You can freeze down your stock of NPCs, and you don't have to do that induction every single time you start spheres. So I've gotten away with very few NPC inductions so far. Chris has done many, many, many more than I have. But I'm also using fewer cell lines than he does. So that's the first thing you have to do. And once you have those stocks, you're kind of good to grow your spheres. The second part, if you want to introduce microglia later to your spheres, you have to basically pick what sort of differentiation protocol you want to use. Though I feel like the world is a little bit divided between the protocols from Sally Cowley's lab. We use an adapted version of her 2022 papers. The other half of the world uses the stem cell kits, basically. Not a big fan of them, but they work too, which we know from our collaborator in China. Those microglia will also infiltrate. So you don't have to use that particular microglia protocol, but it's been working quite well for us. So the way this one works is, yeah, we adapted it a little bit. We use aquawell plates to use these iPSCs and then make them grow these mesodermal embryoid bodies. And those aquawells are nice because, and I'm going to show them soon for the neurospheres too. It's basically a 24 well with like 300 little pockets. And when you spin down your cells, you get 300 uniformly sized EBs or neurospheres. And then you can dislodge them and do whatever. So with these EBs, after a few days, you can harvest those and you put them in those flasks. And we call those macrophage factories. And we keep those for up to 6 months. So those EBs, most of them will attach at the surface. They look quite bizarre, and they are in this hematopoietic differentiation media. And after 40, 50 days, you get these really large macrophage precursors. And those ones you can harvest basically on a weekly basis and then differentiate them further into microglia, or which I will show for the upscaling part, add them straight to your spheres. And that's where you introduce your microglia. So that means you have to consider timing. So how old is my factory? When do you want to start spheres? You can usually use a factory for multiple rounds of spheres, but you have to time that a bit. If your macrophage factory is too young, the differentiation often doesn't work that well. On the other hand, if it's more mature, they differentiate quite well, but the yield goes down. So there's a bit of a trade-off. But that's just what it is. And then this is where we then start growing our spheres. So you basically go to your liquid nitrogen tank, grab your frozen P2 NPCs, you thaw them, and you plate them in that same expansion medium, what we call it. It's the same media at the end of the NPC differentiation protocol, basically. So you plate them for maybe a week until you're happy with the density. And then you harvest those and put them into these aquawells after treating them with a low adherence solution. And if they don't have a chance, if the NPCs don't have a chance to adhere to a surface, they will adhere to each other. And that way form these spheres within 24 hours. So you get these tiny spheres. We keep them in there for like 7 days. And after that, they grow quite a bit, and we move them over to Petri dishes on an orbital shaker. So we switch to BrainFizz at this point, and it's a 3-week long differentiation protocol where we supplement the classic BrainFizz cocktail with cyclic AMP and ascorbic acid. And after that, we remove cyclic AMP and ascorbic acid. And you can keep them on this shaker for quite a while. We kind of settled on keeping them until day 40. And this is where we start plating those spheres. So this protocol, at the end, you plate your neurospheres in 48 well plates. That's what we settled on. I played around with a lot of different plate types, and that ended up being the best compromise for media usage, for workload, for their tissue health. So at that point, when you plate those spheres, I'm going to talk about that in a minute, you also start to harvest your macrophages from your factory and differentiate them on glass for 10 to 14 days into more microglia-like cells. And that's the ones you use in 10 days after. But first, the plating step, that's arguably the most annoying part, because these spheres will attach, and you want them to attach because that's actually necessary for the microglia to infiltrate properly. But how that looks is you have a petri dish with almost 300 spheres, and you have 48 well plates, and there might be 48 well plates with a glass bottom, or here with a cover slip, and you want them to land at a specific spot. So you basically pipette with a whiteboard tip your spheres into your 48 wells, but then you need a microscope to actually position them. That's what we described in detail in the bioprotocols paper, which is actually not that detailed in the Journal of New Information paper. So I was kind of glad we got the opportunity to describe that a bit better. So you kind of have to position them with like a pipette tip and poke them until they land where you want them to land. Then you put them in the incubator and they will attach there. So it's a bit of a workload, but it also has advantages because you have a lot of flexibility on where you want them to grow. Or you can even position several neurospheres on one cover slip with a certain distance to each other, and this sort of things. And then the last step is the addition of microglia. So as I mentioned, you have to differentiate them on glass. So that's part of that Washer et al. 2022 protocol. And then we add them in the last 10 days of the culture. So basically, this is an attached sphere. You see all these processes here. It's over 10 days after plating. And then you add them basically into the media, and they will land all around. And within a certain vicinity, they will sense that sphere moving. And this is kind of a zoomed in image. We use around 35, 40,000 iPSC-derived microglia per sphere in a 48 well plate. And then this is kind of how what we do a lot, and I'm going to show a lot of videos. We do a lot of confocal live imaging. So we have a confocal microscope with an incubator attached and a CO2 supply system. So we can we like to use glass bottom plates where we can always check on our cells. We like to use AAVs to transduce. This, for instance, is synapsin mCherry and GFAP eGFPs. Those were AAV deliveries, so it's very easy to transduce them. And then we stain the microglia with lectin. So this is a far red lectin, and that's just kind of the day after you add the microglia, and you see them like roam around through the sphere. They move in and out, and you get kind of a decent, stable population. Unfortunately, this is limited. They will leave the sphere after like 2 weeks. So that's why we add them in those last 10 days. And that's one big limitation of that protocol. They do not appear to form that really, really stable population. But if you have a really nice batch, this is kind of what you end up with in immunostaining after, when you stain for neurons with MAP2, astrocytes, GFAP, and then the infiltrated microglia with IVO1. You get this really beautiful distribution of microglia, which are, or we're infiltrating into the sphere. Some sit at the edges, and they find their territories like they do in the developing brain. So now I want to talk a bit about the functionality of these spheres. So in a lot of published 3D tissue culture papers, a lot of people unfortunately ignore function. They have their organoids, they do sequencing and stainings, but they don't even show if the neurons are even working, if they're firing. And I like to do a lot of functional imaging, and most of that is, again, AAV driven. So we have a lot of genetically encoded biosensors. We use a lot of GCamp. So if you use Synapse and GCamp AAV, and you add the AAV on the day of plating, you wait 7 days, maybe 1.5 weeks, and you get robust GCamp expression. And then you will see this is an attached sphere, and it's just the spontaneous activity that sphere has. So there's this calcium waves traversing the tissue, and those were reported in the developing cortex and hippocampus. And since those are iPSC-derived neurons, that's developing tissue. And we did a bit of pharmacology to kind of figure out what's contributing to these kind of waves. And of course, you can block them with TTX. They're sensitive to blockade of inotropic glutamate receptors. You can block them with GABA. Conversely, if you block GABA A, receptors, you increase the wave frequency. So you can modulate them with glutamate and GABAergic signaling, basically. And then you can also ask, how do microglia change the activity of my neurons in the spheres? And that's part of the Journal of Neuroinformation paper. So that's a bit of that varies a bit from batch to batch. If you have a batch where without microglia, the sphere was already very, very active, they kind of dial that down a bit. If you have a batch where it's very, very not that active, they will increase the frequency, and the end frequency is always roughly this 0.1 hertz for this particular cell line. So you can ask yourself, okay, how do the microglia modify kind of the neural network? But of course, we wanted to study disease. But before I go into that, we looked at functional properties of microglia. If you know the microglia field, you know that if you burn a hole in the brain with a laser, all the microglia in the surrounding will react and move their processes toward it. And that's a proof of concept that they have the capacity to respond to damage. So we did that, and that's an example of a hole I burned with a 2-photon laser and lectin stained microglia, and you see this microglia sensing it and sends their arms to that lesion. It's not quite a response you get from a mature mammalian brain, but I found that was pretty good already for an iPSC-derived culture. And then, as Chris mentioned, I did a lot of microglia electrophysiology during my time in Professor Ketman's lab. And that was always a way for me to characterize microglia function. So that's what I did for this paper too. I patched microglia, and a healthy physiological microglia should have a resting potential of around -40 millivolt, have a little bit of an inward rectifier, maybe a teeny bit of delayed rectifier activity, but nothing else. So that was matching kind of that physiological state of microglia. And we did a bunch of sequencing. This is based before I go into the sequencing of the pathological model. This is basically just the baseline neurospheres with microglia with a plus or without with a minus. And this is the NUC-seq populations we got. We were really lucky to collaborate with Dr. Frida Miller, who's like an expert in these things and helped us annotate the cell populations. And you see this tiny population of microglia here, which you of course only have if you add them. And then you can answer or test how do my cell population change if I add microglia. And in that paper, we focused a lot on the astrocytes because they changed quite a bit with the addition of microglia, especially in pathology. These are just some feature plots of genes we use to select the different populations or annotate them. So that's the basic protocol. Let's go to the second part, where I want to talk about that disease modeling we did based on this tissue culture protocol. And we use a chronic A-beta treatment paradigm. We originally started with a London mutant line, and the pathology you get from these isn't very striking. You get a little bit of A-beta production, but you don't get neurodegeneration the way it happens in the brain. So we switched to this synthetic A-beta model, and it's a model where we treat for up to 5 weeks. And the beauty is you get these kind of A-beta deposits in the tissue within a few days. And what's interesting about that is that when you look at the function of these spheres with your GChem imaging, they're actually still fine and functional even 2 to 3 weeks into the A-beta treatment, even though you have the deposits already there. And when you think of Alzheimer's disease, where A-beta plaques show up decades before any kind of neurodegeneration is detectable, that was kind of the sequence we wanted to see, that we get deposits, but the pathology kicks in later. As Chris also mentioned, I focus a lot on oxidative stress. I'm going to talk about this sensor a bit more later. That's a redox sensitive GFP. It's a ratiometric sensor, which we also deliver via AAV, and this is kind of the way to show the sensor in this red-green overlay. This is a live imaging of a sphere. Basically, I just image it overnight. And it was sitting in A-beta for several weeks, and I just wanted to catch cells oxidizing and dying in that environment, and that's what we quantified. Basically, there's a higher oxidizing cell rate in the chronic A-beta condition, and if you look at roughly the endpoint of almost 5 weeks treatment. You see basically everything you see, which is green, is like oxidized neural cells. So that's under the synapsin promoter. So these are our neurons. And we can demonstrate that we get A-beta induced oxidative stress, but also that takes 2 to 3 weeks to get. Before that, you don't see anything. So there's that latency where we know the deposits are there, and you need something to break the system to actually get new degeneration. And we wanted to then, another thing that's showcasing the new degeneration. So this is the endpoint after 5 weeks. Those videos are not moving, unfortunately. That's a MAP2 staining, and you can already see how it's much dimmer with A-beta, and the number of processes are lower. We get this dystrophic kind of looking puncta. And so we had pretty clear evidence that a lot of cells were dying. We looked a bit into the cell death pathway. Looks like classic apoptosis is a big one here. So we have a lot of tunnel activity, a lot of caspase-3 activity, hinting that, yeah, these cells undergo apoptosis. And then with this system where the microglia are your optional part, we wanted to see, okay, how do they modify the disease? Because in Alzheimer's disease, it's always like, they can be protective, they can be destructive. It all depends on timing. And that's one thing we noticed with the batches of experiments leading to this one, that timing matters a lot when you add them. So we designed this paradigm where we had a more mild 3-week treatment where we knew we would get oxidative stress and dysfunction on a GChem level without seeing too much death at the end of 3 weeks. And then the more severe 5-week treatment where we know at the end of that period, the neural activity is gone and the oxidative stress is really, really severe. And in those of these cases, we added microglia in the last 10 days and then wanted to see what do they do? How do they react? So the first thing we saw is that there's a lot of gliosis, particularly at the 5-week date. So if you look at just the IVA1 staining here, the number of microglia you get in your spheres in this really severe A-beta treatment is far higher, and it makes sense because a lot of cells are already dead. It will attract them, they proliferate. In the same way, the GFAP immunoreactivity goes up, which indicates astrogliosis. And then we looked at the A-beta levels in this tissue and found the first interesting bit in terms of differences in neuroprotection, that we only see very obvious A-beta clearance if we add them in the milder protocol. So if the A-beta deposits are not that super severe yet, they manage to bring them down. If you do that with a 5-week treatment, they fail to clear that amyloid beta. They do phagocytose a lot, and in fact, we tested like which cell types really phagocytose all that beta, and it's almost exclusively the infiltrating microglia. There's a small percentage of astrocytes participating in clearing A beta, but it's not that much. So even in that 5-week treatment paradigm, they manage to phagocytose, but they get overwhelmed. They will not clear that extracellular A beta that much. And then in terms of function, when we look at the GChem signal, this is a 3-week A beta without microglia, where the calcium waves are basically gone. In the presence of microglia, they're protected. So, and that's the quantification. So this is the reduction of baseline frequency I showed in the beginning when you add them to control spheres. And this is then in the presence of A beta without microglia, where it just dropped, and then with microglia, it got back up. In the 5-week situation, which are example traces here, the neural activity is already down, and microglia failed to recover that. Very similar pattern in terms of oxidative stress. At 3-week time point, you see that increase in this rho-GP ratio. That's the readout of oxidation, and they bring that back down. Now, at the 5-week time point, look at the different scale here compared to this. This is very, very green. There's a bit of a trend, but they don't manage to really help that much at the point. If you look at cell death, though, there's an impact, and you will see that also the sequencing results when we actually look at the surviving populations, they do help in the 5-week treatment group. So there's less cell death, but there's no functional recovery, and that's basically the bottom line here. And this is basically the sequencing results I talked about. So There's a lot here. So basically, we had 6 treatment groups with these experimental paradigms, the 3 groups without the presence of microglia, 3-week A beta, 5-week A beta control, and on the other side, the same pattern with microglia. And I'm going to show a bit of how the populations shift here and how that matches the imaging data we got. So first of all, of course, in the minus groups, we don't have any microglia at all. And then if you remember how we had the massive increase in IVO1 positive microglia in the 5-week group, that's again reflected in the NUC data because most of the microglia are from that particular group. And when you look at neuronal populations, one thing was really striking. There's basically, in the absence of microglia, all the excitatory neurons are wiped out, except for, I believe there's 1 or 2 that survived. All the others are gone. The inhibitory neurons, there are some survivors. The NPCs are pretty much wiped out, and there are some astrocytes. But other than that, it's pretty much dead tissue. And when you add the microglia, you get a recovery. You still have some excitatory neurons, even though we know functionally the spheres are not active anymore, but there is some protection. So at least on like the cell survival level, if not functionally. And then we ask a lot about, okay, how do now the gene expression signatures change? And as I mentioned in the beginning, we focus a lot on astrocytes, and we found a lot of AD-like gene expression in the astrocytes with A beta treatment if microglia were present. At 3 weeks, there were a lot of neuroprotective hits, which were upregulated. Like OXA1 is one which we're particularly interested in. So that's for managing oxidative stress. Then we have CHL1, CCL2. They are all hints that they're going into a protective way. And then one big hit we had with the 5-week was that ApoE. ApoE expression goes up quite a lot, and it absolutely needs the presence of microglia. So you get A beta-induced expression of ApoE in astrocytes, but it needs the presence of microglia. So that was one of the really interesting findings, and which wasn't really known, that you need the presence of microglia to drive ApoE expression in astrocytes, at least to upregulate it. That's similar in a similar way. The same happens to neurons. So this is a subset of the neurons, and also in those, the biggest TEG is ApoE in the 5-week plus neurospheres. So if you have microglia presence with that severe A beta treatment, you get ApoE upregulation in neurons and astrocytes. Although the biggest cell type expressing ApoE is still the microglia. So there's a bit more of ApoE data in that paper I'm not showing here. So it was quite a focus on this story. But I want to go over to the third part, which is the new high throughput protocol we developed, the upscaled neurosphere system. We call it the 384 well system, and the old one, the 48 well system in the lab. So yeah, why did I even move on? So it is a reliable protocol, and like Chris still uses it. He refused to switch over to my upscale system, which also makes sense because it is published. We know the lab in China could reproduce the whole tissue culture. So I was still in the process of figuring this upscaling out, and he was in his PhD thesis and was like, nah, I'm sticking to what's working, which is fair, and it has advantages. And but there are the labor-intensive steps. Like the plating step, the viability, like the spheres after you plate them, they will flatten out. So you can't really go beyond 3 weeks after you plate them. You can push it maybe to 4. And then the microglia tend to leave the sphere after a certain period. Not if they are A beta treated. There was one finding. If there's a pathology, they stick around. If not, they lose interest. So that was a caveat. And a bit of the background of the lab, the long-term goal of all my work was to get a scalable automated model. So we want to have a system where we can eventually screen for mechanisms and drug in a high throughput fashion for AD research. So that was the basic goal and the motivation to go a step further to really scale this up. And that's what I did. And there are a bunch of plates on the market. I actually haven't looked, because I settled on the Corning plates basically. Haven't looked in the last year if there are new manufacturers which advertise spheroid plates. So basically these plates are yeah, I skipped the 96 well part and moved straight to 384. The Acura plates are somewhat fancy, where you have your imaging window, and the window where the sphere forms actually not in the center of the well, which gives you an advantage when you feed them. But the seeding is actually a problem and unreliable. Then you have the Corning and S-Bio plates, where it's basically these tiny round bottom wells, which are a bit different to the Acura, and each of them have their pros and cons, again, like everything. And the Corning plate really won for us for a number of reasons. If you're interested, just email me or ask a question. So now, where this protocol is already so much more advantageous compared to the other in terms of labor, is you basically seed your spheres the same way we do it in the echo wells. We use the same number of cells per well, but we put them straight into these plates, and you don't have to move them anymore. They form these spheres in there, and you can image them in there. You can fix them, collect them for staining, or you collect them for sequencing, or whatever you want to do, but you don't have to move them. You just have those. We leave the outer wells empty for like a PBS evaporation barrier, so you end up getting 308 spheres per plate, but it's like much less labor. But it also came with some tinkering to figure out kind of how to get microglia to infiltrate. Because actually the pre-differentiated microglia on glass, which we do for the 48 system, those don't infiltrate that well into the spheres. The precursors, though, do. And they spontaneously differentiate then into microglia, which looks a little different. And we're characterizing kind of, and that's kind of what I'm doing in the next 2 weeks, to look very closely at their phenotype, their function. At least from the sequencing we have so far, they do look very microglia-like. But yeah, we use the precursors. And this is a very similar video to the beginning, where we can have MJ expressing neurons, EGFP expressing astrocytes, and macrophages stained with lectin. This is, I believe, 2-3 days after adding the macrophages. So you have these very big cells which cling to the edge, and that's where they huddle and cuddle in the first 1 or 2 days, and then start to move in and change shape. So they move from this very big shape to an elongated microglia-like shape. And then this is the sphere which we fixed 30 days after the macrophages infiltrate. And you have that population on the edge, but also infiltrating cells. So this is cryosectioned. It's not a big Z projection. So that's just 10 micron thick. So that's kind of the system you get. But we have the neurons, we have the astrocytes with microglia. The differentiation protocol is very, very similar. What we did notice, though, is even though the differentiation protocol is almost the same, these spheres are very, very different in terms of activity, and that's something I'm exploring right now quite a bit. That's again a 48 well neurosphere, and it has these calcium waves, and we analyzed that back then with Aqua. And we actually set the parameters to just find these big waves, because there's not that much single cell activity. And then when we did the GCAM imaging in the 384 well plates, the activity looks very, very different. So it's this very, very high level of single cell activity, and the calcium waves are actually not that dominant, at least when you have a fairly mature sphere. And so we updated our Aqua parameters to capture both the single cell activity and the waves. So I went back to a bunch of old data and reanalyzed everything to really do a comparison between the tissue culture models. And this is kind of the striking difference, where, first of all, those spheres are younger, but they display this remarkable level of single cell activity at the point where you see almost nothing in the old system. And basically, the calcium wave frequency tends to go down. If you have a younger sphere, they're still there, and then it kind of disappears. And I'm exploring that literally these days with pharmacology, and we seem to have more interneurons, and there's more GABAergic signaling blocking these waves. You can basically turn a switch by blocking GABA A, and then the waves turn back on. So that points to more complex tissue. And we kind of confirmed that a bit with spatial transcriptomics. We're still in the process of going through that, again, with the help of Frida Miller. So another advantage of this system, you can cryosection those. I know Chris tried that with the old system, and it's been challenging because they have all these processes. And when you lift those up and try to put them in OCT, it's really tough to find them again because they float around. Whereas these not so well attached spheres, they're much easier to slice. So we took slices and then did spatial transcriptomics to actually see where are those populations and do we see the same populations we had in the 48-well system. And we do, but the situation is a bit more complex. And yeah, we seem to have a large population of inhibitory neurons. We also have proper OPCs, which was not really the case with the old system based on the nuc-seq data. And then we started to ask, okay, how does the population shift when we add microglia to the equation? And the astrocytes and excitatory neurons don't seem to change, but the inhibitory neuronal population goes down, and interestingly, the OPC population increases significantly. So that's something we have not seen in the 48-well system. So those macrophages, which spontaneously differentiate into microglia-like cells in these plates, seem to drive OPC differentiation. So I think there's potential to get proper oligodendrocytes and myelination, but that will need much longer cultures. And now I'm going to showcase a few things, how we use this and how that upscaling really helped. So we have one collaboration going with Cure, which is a small biotech startup exploring LNP mRNA delivery to the brain to treat pathologies, and they needed a cell culture system to test the LNPs, and jumped on the opportunity to use my spheres. So that's an example where we used a test LNP just to express M-cherry in the spheres. And you get this really striking recordings of a sphere expressing that same GFAP, EGFP. So you see the astrocytes, you don't see the neurons. And then they added the LNPs, and they, like within hours, you see the cells express M-cherry to like substantial degrees. And then they managed to titrate and figure out a half-life of expression. And that was something that would not have worked with the old system because you can't test that many conditions. So it was a nice proof of concept where upscaling then had a huge advantage. And then, of course, we wanted to repeat our chronic amyloidosis model or translate it to an upscaled version, which also needed a few adjustments in terms of A beta concentrations. But we got that to work. So this is again our RoGFP redox sensor. At the bottom, you see A beta treatment. And when we treat these spheres in the 384 well plates, you need to increase the dose a bit, but you do get amyloid deposits, you do get oxidative stress, you see that green signal there. And again, when we add the macrophages, we see that neuroprotective effect. So this isn't as severe as that earlier 5-week treatment paradigm. It's more mild. And again, in that scenario, the macrophages / microglia managed to reduce the oxidative stress, which confirms kind of the data we had from the old system. So, and now we can use that to study drugs. And that's one thing, the last kind of data slide I want to show, where I got interested in the NRF2 pathway, which is this master redox transcription factor, which gained a lot of attention in the AD field. And I wanted to see, what if we tweak NRF2 during this paradigm? Can we actually detect shifts in oxidative stress, and how do the microglia respond? And then suddenly you get a lot of conditions. So we have microglia, you have A beta, you have no microglia, you have no A beta, and then you have your drugs. So this is where these plates really come in handy. And we do semi-automatic imaging. So this is a plate where all the spheres express RoGFP. And then you see this orange to green shift. And that's basically then kind of a color code of the results. And this is a bar graph of the results. So we see, just to guide you through this really quick, these are the control spheres. You add A beta, they oxidize. You add microglia, they go back down. And then if you block NRF2, the A beta toxicity like gets severely exacerbated. And the microglia do rescue a bit, but they fail to do it completely. And NRF2 is supposed to be downregulated in AD patients. The activating nerve 2 didn't do much, unfortunately, so it's probably already saturated, but blocking had a massive impact. So that's just to showcase how this upscaling has a lot of advantages if you want to test different things. So we still don't have a fully automated microscope and fully automated feeding system, but if we had that, we could really scale this up quite a bit and grow thousands at the same time. And that's kind of where we want to go eventually. And yeah, that's what I wanted to share with you. So yeah, big thanks to the three PIs behind this, which is Brian McVicker, Hakan Neigert, and Frida Miller. A big thanks to Ada, who was my undergrad for the past 3 years and helped a lot. Sarah Ebert, who was instrumental in helping us with the NucSig from the Miller Lab. And then, of course, Christopher Lee was a huge help getting that bioprotocol paper written up. And then also Nakita Wenji Kai, who helped writing it. And then another special thanks to Jane, who helped with spatial transcriptomics. And I'm happy to take your questions.
All right. Amazing talk, Stefan, as always. So we have a ton of questions. I've been answering some in the Q&A, and I will try to go through all the ones here. Not necessarily in order. I'll try and have a nice little coherence to them
See the
instead.
chat now.
So I think a big one here, which was asked by Pratib Hayadav. I'm so sorry if I'm saying that incorrectly. What are the challenges you were faced during the process of forming a neurosphere? What are the crucial steps of the whole protocol? So basically the big challenges faced in this protocol.
I guess initially really figuring out what kind of well plates to use. Because before that protocol was written, I tried to basically grow them in 24 well plates and 12 well plates and 6 well plates. And for instance, if you put the spheres after an echo well in a 12 well plate and put them on the shaker, they just bounce against the walls and they will all die. So we needed to find a dish where on an orbital shaker where they rotate in like a nice orbit and actually are happy and survive. So that was a big challenge in the beginning, really figuring out the plate system that works. And then, of course, then the plating itself is always a pain. Yeah, I would say those were the biggest ones.
Okay, so there's a couple on the differentiation here. Deblina asked about the iPSC source. So all of these experiments, or the vast majority that Stefan talked about here, are done using a patient-derived line that we created in lab. But a lot of my work has been done using Jackson Labs iPSC, and these are publicly available to researchers. All you need to look up is Jackson Labs iPSC catalog. I'll probably put a link down here. And we have confirmed on those lines too. They're really appropriate for this protocol.
And also the lab in China used their completely different lines. But the differentiation works, I think, with all of the lines I've seen so far, but the neurospheres do differ though. And that's one thing you also see in the bioprotocol paper. There's one figure showing like the GCamp activity of the 3 different lines. And the calcium waves you get, they do look different between the lines. And it's actually fairly consistent, at least, especially when I see Chris's images using the Jackson line and then mine using the in-house iPSC line. But every batch you see kind of the same pattern, that the activity is a little different. But then also our in-line is female, in-house line is female, whereas the Jackson one is male. So I don't even know if that's a sex difference potentially. But in terms of the differentiation of the spheres, every now and then you have a batch that doesn't really work out, but it's been pretty robust, I say.
Yeah. So another one about the differentiation here. What do you think about the advantages, disadvantages of protocols where neurospheres are formed right after dual SMAD inhibition step, not after a few passages with FGF2 as you do?
One thing we noticed that if you use them at P0, there is a tendency to get kind of dark-looking cell populations, which I think were progenitors, and they were very autofluorescent. So that was the spheres formed, but they often had this kind of populations which made imaging a little funky, and that appeared to kind of go away if you passage them a bit. So the more mature those NPCs are, the better, kind of. That's kind of my response there.
Okay. So this one here from Melissa, if your neurosphere is attached to the plate, the surface attached to the plate surface will not expose to any infiltration of the microglia. I'm guessing this question is talking about how the microglia get into the sphere, considering the bottom of the sphere is adhered to the plate.
Yeah, I've actually seen that in like when I stained for microglia and then for microglia astrocytes and neurons, and then looked at the confocal at the edges of the sphere where you get all these processes kind of like cables connecting to the glass. You see tons of them attached through these processes. And I've seen with our live imaging of the microglia that they crawl along these processes. And I think that's and we had some where the spheres weren't attaching that well. And then that also led to kind of poor infiltration. So I think they need those those processes of neurons and astrocytes to actually crawl along and infiltrate.
Okay. This one from Jelena talking about measuring the oxidative stress. Stefan, I'm just going to link your journal neuroinflammation paper here. We use RhoGFP1 to measure that, a ratiometric marker. Talked in a lot of detail in the J neuroinflammation paper. But Stefan, do you have any other words about that?
I can't find the question. There's so many questions.
It's just about how do you measure the oxidative stress?
Yeah, that's very so it's a complicated sensor because it's excitation ratiometric. It's not an emission ratiometric sensor, which are more common where you have the green and the red protein and you normalize the fluorescence. No, it's actually depending on the oxidation state, the excitation spectrum changes. So that's why you have to use two different wavelengths to excite it, because the oxidized version gives you more fluorescent for one wavelength, the reduced at the other. And that's how you get that ratio to measure oxidative stress. So yeah, I agree with Chris going to that paper or the redox biology paper, or even the original 2004 paper for RhoGFP. So it's a sensor, 2008, which has been around for a while. So there's a lot of, and there are also others which work a little different. So if you are into genetically encoded redox sensors, there are a lot of options. Or shoot me an email if you want to know more.
And Maximilian asked something similar, maybe a bit off topic. How do you perform the AAV transductions? I think that can be really detailed in the J neuroinflammation paper. But anything else you wanted to talk about on that?
Where is that question? What can you read out the question maybe?
Yeah, maybe a bit off topic. How do you perform the AAV
There.
transductions? The calcium imaging in the neurons was already impressive, but especially the AAV transduction of microglia is notoriously difficult.
Oh, yes. So that's where it was a huge advantage working in Brian McVicker's lab before starting to work with Hakon, because Brian used AAVs for his mouse and rat studies a lot. So I did intracranial injections. And then basically when we started to explore this iPSC world, I basically just took the AAVs, added them to the cell culture medium, and it transduces the cells. So there's really no magic, but AAV transduction of microglia is, yes, notoriously difficult. And we have tried a few constructs which were reported to work in the literature, but we did not get them to work. So like robust transduction of microglia, we haven't got it to work. So that's why we use lectin to label them. So if you're really, really focused on microglia, I think so the nice thing here is you could make a mutant iPSC line from your host line and then just differentiate your microglia from that one and the neurospheres from the non-mutant one to have a very specific genetic label only in the microglia. I think that's probably easier than trying to use an AAV to get microglia transduced, in my opinion. Those, yeah, notoriously difficult.
And this one from Dimitri. Small detour question. Are neurospheres generated by a protocol suitable for models of AGEs, related cellular stress? I'm guessing this is advanced glycation end products.
I don't
Which,
know. Not mine.
yeah,
So, but in
it's
general, I think, and that's actually what I would like to do moving forward, especially when I hopefully become independent, to make different model systems based on this, because this is just an AD example, but I think you can model so many diseases or different kind of pathological niches with such a system, especially where you're interested in neuroimmune interaction. So not sure. Maybe.
yeah, it's not something we've looked at.
No.
Jacqueline asks, why do microglia only integrate for about 2 weeks? I think it's a really good question.
Yeah, I have some ideas, and I think it's potentially concentration gradient of the IL-34 or TGF-beta in the microglia media. That they get used to that differentiation media, and we supplement with IL-34 and TGF-beta once we add them, because we tested, like, if you remove TGF-beta, they die. And IL-34 I never dare to remove, but I suspect that if you think you add it to the media, like, the gradient will still be kind of going from outside to inside and from high to low. So I suspect they move in, but in the end they get drawn out again. That's my suspicion, but at the end of the day, I don't know. It might be also that it is potentially more natural to use the macrophage precursors if you think about microglia biology and how they infiltrate during development. And I remember 2 years ago, I had a really nice chat with Florent Ginoux, who is the guy who figured out that microglia are yolk sac derived. And they are aliens of the brain. They move in as macrophages, and then they get shaped by the environment. So maybe by doing that pre-shaping in 2D in a kind of artificial way that you don't get that full integration. But then how well that spontaneous differentiation in the neurosphere works is also another question. So bit of an unknown, I'd say.
All right, I think this one actually is quite important to answer here. What is the optimal age of neurospheres to start the functional studies? Have you measured differences in performance, like the calcium activity of neurospheres of different ages?
Yeah, that's with that published protocol, you're kind of limited to that plating step again. So we have, I think, Chris, you played around more with plating more aged spheres than I have. And
Mhm.
I think you had some mixed results there, right? So because one thing, like, we usually transduce them at the day of plating when they're already day 40. You could add the virus into the petri dish, but then all of your spheres get the same virus. If you want to use different viruses, you kind of have to do it on plating day. So that was easier to explore in the 384 well system, because there I can just transduce them earlier because they just sit in these plates. That's also why when you look at the GChem traces where I compare them, I start way earlier in the 384 well system because I can. I don't think I've ever tried to plate really, really young spheres, because one thing we notice is that the GFAP expression kicks in around day 40, day 50, and we wanted to have astrocytes. So we kind of didn't dare to go younger. At the end, it kind of depends what you want to do. If you don't need or want mature-ish astrocytes, you can probably go earlier. In terms of later, I'm not sure. I was always curious to maybe leave spheres in these petri dishes for a month and then try to plate them. I've never really tried that because at the end I moved over to the upscaling part. So I'm not quite sure. We know
Yeah,
that
I can.
works, basically, that's published. If you deviate from it, Might work, might not. So maybe Chris has some comments there too, because I think he played around more with that.
Yeah, so in terms of like the start of our calcium expression, the earliest I've gotten have been that using this protocol that we're kind of talking about here has been that day 44 after the neurosphere formation. But I have used put them on the MEA and gotten, the MEA for us is we've done MEA side by side with calcium imaging. We find it lines up very closely. Calcium imaging just gives us a bit more um, consistency there. For example, if we put the sphere on the MEA plate and it just misses an electrode, because the spheres are quite small, depending on the plate, you might just not get any activity. So, but we have looked at them from an earlier stage, kind of around day 40, and seen good activity. So my suspicion is that they will be active even earlier. It again, like Stefan said, just depends on what you're looking at. And I have also plated spheres up to day 90. But one thing I found, and this is similar to just our differentiation from our NPC protocol as well, is after day 60, they sort of tend to decline. That's kind of been the endpoint for a lot of our experiments because of that. So plating these spheres at day 90, they're quite a bit, quite unhappy compared to just if you're plating them earlier. So let me see where are we on the questions here? Fanny asked about the stages of the microglia. So in respect to microglia function, are they developing into dam-like stages, or how diverse are they in the model where you added to the A beta over time?
Yeah, that's actually in that paper. I don't have that in the presentation here. And that's one thing the reviewer asked, like, how do the microglia change with A beta treatment compared to controls? And that's something we couldn't really answer because we had a lot of microglia or like a good number for that 5-week treatment because there were so many more. You remember that IBA1 staining. So that population was pretty large, but then the controls, it was just, I don't know, 20, 30 microglia. So we couldn't really do a comparison, but we did dissect the microglia from that 5-week treatment group and looked at how they subclustered. And basically, we found that they do separate into 3 different stages. We get a dam-like expression, we get a more macrophage-like, and we get a more homeostatic. How that is triggered by A beta is unknown because we can't really compare with the controls because there's so few cells, and we haven't done more nuc-seq runs since. So that's one thing I hope to understand from the spatial transcriptomics, but we're not there yet.
Okay, and I think this one was asked before you started talking about your OPC cells, but this one by Federica. I noticed that oligodendrocytes are absent in both your organoid models and the RNA-seq data. I think she meant spheroid models there.
Yeah.
Is this due to a limitation in the protocol's maturation timeline, or is it technical bias in cell capture?
I think it's the timeline. And I think that if we go back to here, this population here, the glial precursors, which Frida actually annotated them as that's a small population which expresses OLIG1 and OLIG2. So those could be called OPCs, but they don't express markers for proper oligodendrocytes. And what we then saw in the spatial transcriptomics that OPC population seems larger, especially when we add microglia, which is another hint that we think they mature faster in these smaller wells, together with the functional data. So yeah, getting oligodendrocytes in these spheres, I'm not sure what the best strategy would be to kind of try to increase cell culture time. I think if you would do daily feeding, you could probably stretch it quite a bit more, or kind of try to differentiate oligodendrocytes in parallel and add them to the culture similar to how we do with the microglia. I don't know which strategy would be best. I haven't focused on oligodendrocytes much at all in my career, but been seeing a lot of interest in that. Also, when I show this stuff on posters, everybody asks about oligodendrocytes. And like, I'm not sure. I think the capacity is there because we get the OPCs, but it needs more, either longer cultures or you, yeah, differentiate them in parallel and then add them. But I don't even know if they would infiltrate, how they would interact. That I don't know.
Okay, let's see, where are we now? And Renee, how much time do we have here?
We are flexible. It's up to you guys.
Okay, we'll try and go through all the questions here then. I think we're making our way. So you answered this. This was a bit at the start of your talk, but maybe this person missed it. Would be good to mention again. So thank you for the explanation. What are the advantages of neurospheres compared to organoids, considering that organoids better mimic native tissue?
Yeah, it's size, experimental control, reproducibility. So I've seen way more variability between organoid batches than sphere batches. And then if you want to integrate microglia, you have to deal with that necrotic core, which is, I think the field gotten better, and there are protocols where the necrotic core is really limited. Even spheres can develop small necrotic cores, but they're not to the point that the microglia go for it and just eat up the whole sphere. Yeah. So, and then the organoid differentiation, yeah, is more complex, but also less plannable, I guess. It's in terms of what you get, what kind of brain region or early brain regions form at what spot in the, or it depends on your research question. If you need that complex tissue, if you look at microglia impact on cortical layer development in human system, you need to use an organoid system. It depends on your question. So
Yeah, I think that is very, very true. Really, it's just, these spheres are one part of the research options out there. We're not saying that these are
yeah,
at all better than organoids, but
yeah,
it fits a niche,
it
and that's
depends
really what
on
we're
your
aimed
question.
at.
Yeah.
Yeah. Similar to this reproducibility, do you have any data on the level of transcriptome variation between different neurospheres from the same batch or different batches?
We only have two different batches of nuc data, and that variability was fairly limited. The one difference was actually the number of microglia captured in one batch was way higher than in the other. But we haven't explored that too much. But I know that's something Hakon really wants to do, and like actually like sequence at different time points and really see how the transcriptome changes over time. Yeah, but we don't have that data yet.
Can endothelial cells infiltrate the neurospheres and form BBB-like properties? That's a really good
I
question.
have no idea, but we're actually going to try that in 2 weeks. So I don't know. I had some pilot fun experiments with somebody who was growing uh, vascular organoids, and basically you chop them to pieces and then add those vascular pieces, and we added them to the spheres. But those pieces, basically the vascular pieces, were larger than the spheres, and it was kind of a interesting experiment. And basically what we saw was massive GFAP upregulation in the spheres, so a lot of reaction from the spheres. And the sphere was kind of migrating towards the vessel rather than the other way around. So it looked very bizarre. But by just adding kind of single cell endothelial cells, like a single cell suspension, we haven't done that yet. Same with like parasites, smooth muscle cells. We haven't done that. I don't know. So I honestly am very skeptical regarding BBB models with these 3D tissue cultures, because I always think it needs a heart, a pumping vasculature, and that's just incredibly difficult to model, especially when you want to scale it up. So this model just doesn't have that vascular component. And again, the research question matters if you are into vasculature. That needs a different approach. But on the other hand, we haven't tested much. So we're going to explore that a bit in the next few weeks. But again, will they form really vessels if there's no connected pump? I'm unsure. So how complex that interaction will be, we don't know. But I know there are some papers where they got pretty nice BBB-like formations in spheroid cultures, like the one fairly recent paper where it's literally like a barrier around the whole sphere. And then you treat outside as the vasculature and inside as the brain, and then you can look at Evans blue transfer and something like leakage. Maybe that would work, I think, but I wouldn't go beyond that with my expectations.
Yeah, and I think that the nice thing about perhaps the 48 well model or this Petri dish model is that compared to maybe the upscaled models is that they might be more malleable to adjusting to other protocols. For example, if you're wanting to look at microfluidics, you can very easily pick out these spheres and transfer them over. So I'll move on to the Q&A questions now because there's a few more over there. I did want to say though, and Stefan will echo this, I'm sure, but thank you to everyone for tuning in from around the world. It really is quite incredible. And if Stefan, you had any words on there I just want to say before we lose too many people, as we're still going on the questions.
Agreed. Thank you. Oh, there's a Q&A. I found it. Good.
Yeah. So we have TechnoKL5 asking, in this context, is the macrophage the same with bacteria virus? So I'm
not sure I understand the question.
Yeah. I'm guessing it's just asking about like bacteria infiltrating into the spheres, perhaps. Not entirely certain.
Yeah, I've never done that intentionally, unless we had contamination. I'm not sure. And also, since we're not really interested in bacteria or bacterial viruses, I've only experienced or have experience with AAVs as a transduction system. We haven't even tried lentiviruses, so I'm not quite sure.
Jacqueline H asked, how do you know if there's any necrosis in the center of the neurosphere? I think you answered this a bit already, but did you want to say anything else on that?
One thing that RoGFP sensor is really good for that, because if there is a necrotic core, you will get oxidation in the core. And you will also see that the infiltrating microglia accumulate around it. That's where you can control, and it's again an advantage over the organoids, you can control the size a bit better. And that's one difference we do in the 384 well system, that 7-day expansion phase in the echo well plate determines a lot how big your sphere will be at the end. And if you cut that shorter, they get a little smaller. That's how you can control it. So you don't want a sphere with a diameter larger than 500 microns, because then you will get these small necrotic cores. So I've seen it. You can even sometimes see it in transmitted light, but like for me, like the RoGFP expression, like seeing that oxidative damage is where I spotted right away.
I think that's actually something I wanted to chime in on as well, that the early stages of the agar well, those are really critical days to your sphere. And a nice thing about this protocol as well is you can sort of identify early if something has gone wrong, because you'll see it in the agar well before you move it over into the Petri dish. Usually, if they're nice in the agar well, if they're of good size, of good shape, they will be good in the Petri dish. Hemlata asked about the presentation. Hi, thanks for the beautiful presentation. Your PowerPoint was extremely beautiful. Could you please tell me how to learn and proceed to design such an impressive PowerPoint? Thanks in advance.
Well, thank you for the compliment. Practice, right? Give as many presentations as you can, wherever you can, and get feedback. And yeah, thank you.
And Pratibhanya, this protocol will be the same that we can use for the sphere information for the Microbacterium tuberculosis. It's another bacteria question.
But
Again, not too sure.
but it is not a question though, it's a statement.
Yeah. So
Uh,
not too sure about this one.
Same.
Um,
Or the next one, the caspase-3 and nerve2.
yes.
Yeah, caspase-3 is part of classic canonical apoptosis, and it gets cleaved during that process. So imaging cleaved caspase-3 is basically a marker for apoptosis. And then we combine that with tunnel, which is another marker. And nerve2, as I said, is a transcription factor. So it's a master regulator of an oxidative stress response. So that transcription factor basically sits in a cytosol or in the cell membrane, and it basically gets released by oxidative stress and translocates to the nucleus, and then it ramps up a bunch of oxidative stress gene expression responses. So that's why I like that to manipulate my redox situation, because I didn't touch a specific gene. I just switched a whole transcription factor, which should target a lot of different things, to just see differences first. So yeah.
So this one from Miriam, I think we answered that already, the neurospheres versus organoids,
Yes.
but she has another there. When you're optimizing for growth conditions such as the type or of plate or medium composition, what are your readouts? Do they rely mostly on the morphology of the spheres, or would you look at marker expressions?
Mm, I mean, we really haven't played much with medium composition, right? It's in terms of plate, it was again morphology of spheres and also my redox indicator. Basically, when I described that spheres bouncing around in a 24-well plate or 12-well plate, you could literally see the outside of the sphere being oxidized because it was bumping against the wall. And then I know, okay, this isn't working. Um, yeah, but I'm sure there are different markers you could use to do that in a more systematic fashion. Um,
Yeah, I think media composition as well is something that is not anything we've looked into. Really, this media we use is very similar to the one we used for our 2D
yeah.
neurons that our lab has been doing for years and years. We sort of just transfer that over to 3D, and it worked very well. So there's a huge opportunity there for testing
Yes,
different medias.
it's just very, very tedious work.
Hmm.
Um,
But a opportunity there.
yep.
What is by Yazid? Quick question. What is the rate of forming the
Yeah.
neurosphere? I
Well,
assume that's what
that's
that's asking.
the rate in terms of growth rate. I mean, that's kind of the whole overview in the beginning, the 60-day protocol we have for the 48-well system, and I do roughly 50 days for the upscale system. Or the rate in terms of how many batches you do, I'm not sure. Um, like,
Uh, in terms, I mean, maybe it's asking about like NPC to sphere amount, and that we can quickly say
oh,
that
I
we start
see.
with like one, maybe 1.5 million, and get around like
So we
300
use
spheres.
3,000 NPCs, I believe, if I 3 or 4,000, I should know that, per sphere. Um, so yeah, you basically grow your NPCs until you have a confluency where you're confident you have enough for how many echo wheels you want to seed. Um, yeah, and that's it.
But yeah, if you had more specifics on that, feel free to type another
Oh,
question there because
that
we're
last
just at the
question
end
is a
here.
very, very good one from Fanny. Um, So the lectin, I was super happy that it actually stays around for a week at least. It gets a little fuzzy because it sticks to the membrane, right? So at the end, you don't see processes that well anymore, but you can see the cell bodies of microglia for up to 2 weeks. In terms of when to apply it, that was kind of my first attempt were very sad because I added the lectin after I added the microglia to spheres without knowing that there's a large body of literature from the 70s and 80s describing how neurotoxic lectin is. And all my spheres died after 24 hours. And initially I thought my microglia are killing my spheres. And at the end, it was just the lectin. So what we do is we add the lectin to the microglia cultures 30 minutes before we collect them, and then we spin them down, resuspend in new media, and then add them to the spheres, and then that toxicity is not an issue anymore. But like I know the lab in China use a different dye, so you can use pretty much any live dye. But then I don't know which one. If you have one which is particularly known to stick around for a long time, that might be even much better than lectin. But lectin we use because it's known to bind to microglia membranes.
Great. And then one last one here from Samson. For the sake of students from developing countries, especially in Africa, how can your organization extend the training or have a center where the students can have an understanding to carry out research in cell biology in Africa? Is there a way such training can be extended? Because many lack these skills, theory, practical aspects of this discipline might be beyond the scope
That's a
for you,
toughie.
Stefan, but
A toughie and a
but it's an
political
important
question.
It's a
It's an
really
important
good
question. I
point.
totally agree. And I mean, one thing is uh, one issue with these techniques is also costs. Like these iPSC-derived cultures are really expensive. So setting up like an organization or like training is probably challenging. So yeah, I don't really know how to answer that one, but I totally agree. That's an important one. Um, yeah, I
Yeah,
can
I know.
imagine it's challenging in terms of resources. We were fortunate that we
Mhm.
had a lot of private donor funding from private donors who wanted to fund Alzheimer's research, which got this going. Um, yeah.
Yeah, and that seems to be the last of the questions.
All right.
Um,
That was a lot of them. I like it.
yeah, that was great. So, of course, if you have more questions, Stefan, do you want to switch over to your email again or put it in the
Just email
chat?
me. We're happy to help. And yeah, I've been already contacted by one or two after they saw, I don't know, the JNU information paper or bioprotocol. If you want to really establish it and want kind of us as a contact to be there for questions, just email us. I think, Chris, your email is also on the bioprotocol paper, right? As contact. I think both
Yes.
are. So we are happy to guide you through it if you stumble across any kind of complications. Maybe on that note, it's actually interesting story when the lab in China started the neurosphere protocol, and I actually flew over for the talk and also help a bit in the lab. And the student there, I don't know if she's online, um, kept saying that her neurospheres in the petri dish would get fewer and fewer. So she was concerned that she was aspirating them when she was feeding them. And at the end, what the issue was that it was a different type of petri dish, which was, I think, a little wider and less, and the height was different. And on her shaker, basically, the spheres would eventually every now and then touch the wall and stick to it. So I saw like a smear at the edge, and all it was a different type of petri dish. So that's something that's just so difficult to address. And when you write this kind of paper, like, but those are random pitfalls. So if you try to establish that in your lab and something is not working, just email us. I'm sure we can help you out. So yeah, don't hesitate.
Great. Well, thank you so much. I think I might hand it back over to Renee if you have